Hello, everyone, and thank you for joining us for part 2 of our 3-part immuno-oncology series. Today's live broadcast, Advancing Precision Immunotherapy Through Next-Generation Sequencing of T-Cell Receptors, Part 1, is presented by Dr. Timothy Looney, Staff Scientist, Bioinformatics, Thermo Fisher Scientific. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational webcast is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Answer Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd now like to introduce our presenter, Dr. Timothy Looney. Dr. Looney is an accomplished computational biologist and immunologist with expertise in methods and applications of B and T cell receptor sequencing, including biomarker discovery and assay development. He is currently a staff scientist and informatics lead for immune repertoire sequencing within the Clinical Sequencing Division at Thermo Fisher Scientific. Dr. Looney's interests and specialties are centered around the repertoire analysis for immuno-oncology, autoimmunity, and infectious disease translational research. Dr. Looney, you may now begin your presentation.
Great. Thank you for that very nice introduction. As mentioned, I'm going to talk to you today about the sequencing of T-cell receptors. And if you tuned in for the previous installment of this webinar series, you may have been introduced to our immuno-oncology portfolio. There are 4 assays within this immuno-oncology portfolio. The Oncomine Immune Response Research Assay, which is a targeted gene expression profiling assay meant to allow one to understand the tumor microenvironment. There's the Oncomine Tumor Mutation Load Assay for profiling tumor mutation. And then what I will focus on today, T-cell receptor sequencing, in particular our Oncomine TCR beta LR assay. The LR stands for long read. And in the next installment of this webinar series, I'll discuss the TCR beta SR assay, which is our short read assay compatible with FFPE preserved materials. So to put everything in context in our broader strategy in immuno-oncology, I want to introduce the concept of checkpoint blockade immunotherapy. And this is an incredibly exciting development for the treatment of cancer. Checkpoint blockade immunotherapy describes the use of agents that modulate the way in which T cells interact with immune cells in tumor in order to increase T cell killing of tumor. When it works, it's incredible. There's durable response, and in some cases, an individual with cancer is totally cured. What is potentially even more exciting is the notion that one can use these blockade agents in combination to further increase the efficacy of this immunotherapy treatment. So one question is whether or not someone will respond to an immunotherapy. This is something, predicting response is something that is an outstanding question in the field. Now, despite the efficacy and excitement with this approach, there is a challenge, which is immune-mediated adverse events. And when one receives checkpoint blockade therapy, that can activate T cells to destroy cancer, but it can also make those same T cells destroy healthy tissue. And immune-mediated adverse events or immune-related adverse events can affect multiple organ systems and cause permanent damage or even death. And what's more is that the incidence of adverse events is elevated in combination immunotherapy setting. Thus, the therapeutic approach that holds the most promise may also have the greatest challenge in terms of toxicity. And to date, there isn't a good way to predict who might have a very bad reaction to immunotherapy and who might receive checkpoint blockade without problem. So if one could identify biomarkers, predictive biomarkers for adverse events, that would allow for safer and more effective use of immunotherapies, potentially allowing for personalized drug selection and dosing, and ultimately expanded use of the most efficacious combination therapies. So at Thermo Fisher, I want to mention that we have taken these questions to heart. And much of our work has been focused on identifying predictive biomarkers for adverse events during immunotherapy, but also predictive biomarkers for response. And in the following slides in this webinar, I hope to show you how T-cell receptor sequencing of peripheral blood can be used to address both of these questions. Now, in terms of the immuno-oncology assay portfolio, this portfolio was designed in order to interrogate all aspects of the cancer immunity cycle. And this details the way in which T cells and other immune cells interact with cancer. In the diagram here, you'll note at the bottom a visualization of the tumor microenvironment. And the tumor microenvironment is a perfect type of material to interrogate with the Oncomine Immune Response Research Assay, which is a targeted gene expression profiling assay. Tumor may also be profiled using the Oncomine Tumor Mutation Load Assay. And antigen-presenting cells may prime T cells with tumor neoantigen, which may proliferate and then ultimately kill tumor. I'll point out to you in this immunity cycle the Oncomine TCR beta LR assay. It's on the peripheral blood portion of the immunity cycle, and this assay is really ideal for interrogating T cells within the peripheral blood. These are T cells that may have been primed by tumor neoantigen and proliferated. And so within the periphery, one may detect signatures of T cell responses to tumor. So in this next slide, I'm giving a little bit more detail in the T cell receptor biology. The T cell receptor is a heterodimer consisting of a beta chain and an alpha chain, which are produced by VDJ recombination. And during this process, tandemly arranged variable diversity and joining genes recombine to produce a functional receptor. Owing to the combinatorial nature of that process and the action of exonucleotide chewback at the ends of the V, D, and J junctions and addition of non-templated bases by TDT, the CDR3 region is incredibly diverse. It's so diverse that it can be used to track a T cell over time. With that said, the CDR1 and 2 regions of the rearranged receptor are also important for determining the antigen specificity of the receptor. And this portion of the receptor is germline encoded. And what I'm going to describe to you in the later slides is how polymorphism within this germline encoded portion of the variable gene may serve as a biomarker for adverse events. So this slide highlights the assay design of our TCR beta chain sequencing assays. We have developed an LR and SR assay. LR refers to long read, and it's a long amplicon, and SR, short read, short amplicon, covering the CDR3. Now, the CDR3 only assay, the short read assay, owing to that short amplicon length, allows it to be compatible with FFPE preserved material where the nucleic acids may be degraded. By contrast, the LR assay, you'll note at the top, utilizes multiplex primers targeting the framework 1 region of the variable gene and the constant gene region. So this produces an amplicon of approximately 330 base pairs in length that includes sequencing coverage of all 3 CDR domains. Not just the CDR3, which can be used to track cells and measure clonal diversity, but also the germline-encoded CDR1 and 2 regions of the receptor. Now, both of these assays can be analyzed in Ion Reporter, which is our analysis platform, and this includes a number of analyses that allow one to quickly get a sense of immune repertoire features. This includes visualizations of the repertoire, which allow one to quickly get a sense of the extent of clonal expansion or diversity. The software allows one to track clones over time, so longitudinal tracking of clones. And I'll note that the data produced by this assay is freely available to customers. You have access to the raw data, and this analysis can be run locally on your Ion Reporter server or cloud. So, apart from the analysis, which is a great part of this tool, we did a lot of work to ensure that the assay is producing accurate and sensitive readout of the clones within a sample. And one way we did this is by creating a set of reference rearrangements derived from literature. Here I note the details of this procedure, where we used 30 TCR beta chains derived from T-cell lymphoma cell lines. The 30 that I mentioned were actually used to validate the Biomed2 TCR beta primer set. We took the sequences of these beta chains, cloned them into plasmids, and then used those plasmids to spike in to PBL, peripheral blood leukocyte background, in order to assess the sensitivity of this assay. Now, in this slide, you'll note the results of that experiment. On the left, I'm showing you the results of spiking in those 30 reference rearrangements into a background of cDNA derived from peripheral blood leukocytes, and asking whether or not one can detect those reference rearrangements over a range of input frequencies. And so you'll note that the frequency of these reference rearrangements matches expectation, and the assay can detect rearrangements at over a wide range of clonal frequencies. Now, on the right, you'll note a complementary experiment to demonstrate the accuracy of this assay. In this experiment, we sorted a defined number of T cells, extracted all of the RNA from those T cells, and then used that for library preparation. Now, in this case, you don't know the sequence of the rearrangements, but you know the maximum number of rearrangements that one might detect. For example, if one sorts 5,000 T cells, extracts all of the RNA, and then sequences the TCR beta chains, one shouldn't expect this to find far more than 5,000 unique rearrangements. If one did, that might indicate that there are errors or artifactual rearrangements owing to sequencing or PCR error. So what you'll note here is that over a wide range of T-cell input amount, the assay reports a number of unique rearrangements or clones detected, closely matching expectation. So now I'm going to switch into the biomarker applications, and I think this is a really exciting portion of the talk. After I cover biomarker, I'm going to then discuss applications of this assay to T-cell therapy. Now, this slide highlights two important biomarkers that one can obtain from using the TCR beta LR assay on peripheral blood. And you'll note on the left is the results of centrifuging peripheral blood. You'll note the plasma fraction, the buffy coat, which is the white blood cells where the T and B cells can be found, and then the erythrocytes. Now, when we typically think about liquid biopsy, we think about sequencing material within the plasma fraction. And the buffy coat may be ignored. Now, what one can do with this assay is extract total RNA from that buffy coat and use that RNA as input for library preparation through the LR assay. And that allows one to do two things. Measure TCR convergence, which may be a biomarker of, a predictive biomarker of clinical response, and I'll talk about that more, but also assess variable gene polymorphism. And there's a thought in the literature that polymorphism within the germline encoded portion of the variable gene may be linked to autoimmune disease and potentially toxicity during immunotherapy. So just a reminder of the LR assay and the amplification strategy. As I previously stated, this uses multiplex PCR primers targeting the FR1 region of the variable gene and the constant gene region. That allows one to detect polymorphism within the variable gene, but also assess the CDR3 region. And I'm highlighting right now the domains of this amplicon and how they may be used. Polymorphism within the germline encoded portion, and in the CDR3 region is essential for measuring TCR convergence, which refers to the phenomenon whereby T-cell receptors are identical in amino acid space, but distinct in nucleotide space. So a typical use case of this assay is 25 nanograms of cDNA derived from peripheral blood total RNA, sequencing the resultant libraries to 1.5 million reads depth, and that turns out to be about 8 samples per 530 chip. And I want to mention that the reason we are able to sequence such a long amplicon is owing to the fact that the Ion Gene Studio S5 530 chip allows for quite long read lengths, up to 600 base pairs. So this assay is really, you could argue, is enabled by this unique technology that the 530 chip provides. So in the next slides, I'm going to show you some data demonstrating that these two biomarkers may be used to predict response. And the data I will present to you is derived from 55 individuals who received checkpoint blockade monotherapy for cancer. They received either ipilimumab, nivolumab, or pembrolizumab. These were almost entirely Caucasian. 54 were annotated as Caucasians and one of unknown ethnicity. And the sample was pre-treatment peripheral blood, 25 nanograms of cDNA, and the resultant libraries were sequenced to 1.5 million reads depth using the S5 530 chip. And this experiment was performed through a third-party OmniSeq, and the samples derived from Roswell Park Cancer Center. And to the right, I'm showing you a breakdown of this, the details of this cohort. You'll note that there's a range of cancers, mostly melanoma and adenocarcinoma, but some others. And then at the bottom, repertoire features. This highlights just the fact that the samples were sequenced. The sequencing quality was good. We obtained about 1.5 million raw reads per library. So to talk about this polymorphism concept, I also want to bring into discussion the concept of a haplotype. And a haplotype is a set of DNA variations or polymorphisms that tend to be co-inherited. And this can be owing to genetic linkage or population structure. Now, the TCR beta locus is a classic example of where one would expect to detect a haplotype type structure, because there are polymorphic variable genes which are in close genetic proximity. And because this assay allows us to detect all of the variable gene alleles within an individual's repertoire, we can infer the haplotype from the data generated by this assay. And thus we can ask whether particular haplotypes are associated with severe adverse events during immunotherapy. And in this slide, I'm highlighting a cartoon example of two different haplotypes, where the boxes indicate genes, the star indicates different variable gene alleles, and the color indicates what the actual allele. And you'll note that with a haplotype, one expects to see sets of co-inherited or coincident alleles. So in order to determine the variable gene alleles present in an individual, it's actually quite straightforward from the standard ion reporter analysis workflow. And after the FR1 framework 1 constant gene multiplex PCR, there's sequencing of the resultant amplicons. There's a step to eliminate PCR and sequencing errors, and this takes advantage of the fact that the ion torrent has a very low substitution error rate, and substitution errors are problematic for immune repertoire studies owing to the fact that they can mimic the natural variation in the repertoire. After that error elimination step, there's an annotation step where the rearrangements are annotated by comparison to the IMGT database. Now, IMGT database is considered by many to be gold standard, but it's nonetheless thought to be incomplete. And thus, when annotating by comparison to the IMGT database, one also has to have a procedure to identify instances where an individual may have an allele that's not in the IMGT database. So we can really take advantage of this long amplicon approach where the germline encoded portion of the variable gene is included in the amplicon along with the CDR3. And in instances where an individual has an allele that's not in the IMGT database, it will manifest as a plurality of clones having a systematic mismatch to the IMGT database over the variable gene portion. So multiple clones, each with a readily distinguishable CDR3 nucleotide sequence, all having the same mismatch to reference. And we know if we observe a plurality of clones having a mismatch, then that indicates that there must have also been a plurality of unique template molecules representing the non-IMGT allele. So in looking at this data, in addition to determining all of the IMGT alleles an individual has, we can also determine instances where there is a non-IMGT allele. So applying this methodology to our 55 individual cohort, we obtain something like this. This heat map on the right indicates the distribution of alleles across individuals. This is an allele profile of each individual. Now, in the heat map, each row indicates a different individual, and each column a different allele. A red tile indicates that an allele was detected in an individual, and a blue tile that it was absent. So you'll note firstly that some columns have all red tiles. This is an example of a ubiquitous allele. Everyone in the cohort had that variable gene allele. Now, other columns have mostly blue tiles. This is an example of a rarer allele. Now, you'll also note this box-like structure, this blocks of tiles, red tiles that tend to be found together. This is a manifestation of the haplotype structure that I mentioned. And on the left, I've subdivided the data into 4 major haplotype groups based on the patterns of these co-incident variable gene alleles. Now, in the next slide, what I'm going to do is project this data into 2 dimensions using principal component analysis. And then I'm going to overlay that projection with the adverse events annotation of the cohort. And here it is. You'll note on the right, each symbol represents a different individual, and each color a different haplotype group. And the symbol shape indicates the grade of the adverse events from grades 1 to 4. And you'll note the red haplotype group. This group makes up about 30% of this Caucasian cohort, and no one in that group had a grade 3 or higher adverse event. Thus, they appear to be protected from severe adverse events. Now, this could have implications for the way in which one might theoretically one might treat these people. This haplotype group 2 may be more amenable to a combination immunotherapy approach where there is higher toxicity. They might have just a very low risk of toxicity. They also might be able to receive a higher dose of monotherapy agents. And then one can also ask the question whether or not this haplotype group 2 cohort may be at lower risk of chronic autoimmune disease. So certainly, if you have a question, please write it down. Contact our moderator, and we'll review these at the end of this webinar. So I'm gonna leave it at this for the haplotype and polymorphism. The one other thing I just want to emphasize is that these haplotypes are germline encoded, so one can assess the haplotype of an individual at any point in time. It's not as though one needs to assess it at the time of a toxicity. So in that way, it can potentially serve as a true predictive biomarker for adverse events. Now, the second concept I mentioned before and I want to discuss in detail is TCR convergence. And this refers to the phenomenon whereby an antigen stimulation event may give rise to T cell clones which are identical in amino acid space but different in nucleotide space. And in such instances, we know that T cells arose, independently went through VDJ recombination, and proliferated in response to some common antigen. This is what the Convergent TCRs refers to. And one of the really interesting things about convergence is that it may preferentially arise owing to chronic antigen stimulation rather than acute but transient antigen challenge. And the reasoning is that in chronic antigen stimulation, the pool of T cells that have a chance to be stimulated by the antigen is far greater in the context of chronic antigen stimulation than acute but transient antigen stimulation. And for that reason, convergence may be a great way to get a sense of the immunogenicity of a tumor, and thus the likelihood that it may respond to immunotherapy. So I just want to give you an example of what a convergent TCR group looks like. Here I'm showing you an example of a convergent group, 3 clones detected in an individual with melanoma, and this is in the pre-treatment peripheral blood of an individual. And you'll note that these 3 clones are identical in amino acid space. They have the same variable gene, the same CDR3 amino acid sequence, but the CDR3 nucleotide sequences are different. And they differ at the VD and DJ junctions at the part of the rearrangement that arises owing to exonucleotide chewback and N addition by the enzyme TDT. And if you look carefully at this slide, at the black bases adjacent to the blue and the yellow portion of the CDR3 nucleotide, you'll note that a single base substitution, a substitution sequencing error, has the potential to create an artifact that resembles a convergent TCR group. So for this reason, substitution sequencing errors are particularly problematic for the measurements of convergence. And getting back to Ion Torrent and the advantages of this platform, Ion Torrent has a very low substitution sequencing error rate, and thus it may be particularly well suited to the measurement of convergence. Now, we have been looking at convergence as a biomarker for some time, but one of the first places we looked was through the sequencing of tumor biopsies. And here I'm showing one slide from data we presented at AACR of this year. And this study involved sequencing of the tumor-infiltrating T-cell melanoma. And this was using the LR assay, and the input samples were fresh frozen tumor biopsies. And one thing we noted is that within the melanoma-infiltrating T-cell repertoire, Convergence, we found ample evidence of convergent TCRs. Quite different than what one observes from the sequencing of peripheral blood from healthy individuals. So one question we then asked was whether convergence might also be detectable in the peripheral blood and whether it could be used as a biomarker to predict response. So now returning to that 55 sample cohort, when we looked at the pretreatment peripheral blood for evidence of convergence, we noted that indeed it appears that individuals who go on to have an objective response tend to have a higher frequency of convergent TCRs in the pretreatment peripheral blood than those who do not. And this emphasizes the fact that the peripheral blood, T cells in the peripheral blood, may indeed hold signatures of T cell responses to tumor, and thus can serve as a liquid biopsy type material for predicting immunotherapy response. So in terms of what I just showed you with respect to biomarkers for TCR, I've talked to you about convergence and prediction of adverse events. Now, one question is how convergence might relate to other biomarkers. For example, tumor mutation burden. And this question is interesting. Now, one can make the argument that in some ways, tumor mutation burden may be an indirect measurement of the immunogenicity of a tumor, because the somatic mutation must lead to a change in amino acid of an expressed protein, that a peptide must be presented by the HLA, and then ultimately has to engender a T-cell proliferation and killing of tumor. So there are many steps that have to occur between the somatic mutation and the T-cell response. But with convergence, we're actually measuring the active agent, which is the T-cells themselves. So there is an argument to be made that potentially something like convergence may be a more direct measurement of tumor immunogenicity. And finally, just to emphasize, the data I showed you from pretreatment peripheral blood allows for both assessment of polymorphism for prediction of adverse events and convergence. So with a single library, one can address these two important questions. So now switching to the last section of this talk, T-cell therapy, I'm going to talk to you about ways in which this assay may be used to improve the manufacture and design of therapeutic T-cells. And the use cases can be subdivided into two major categories highlighted on this slide. First major use case for TCR sequencing in therapeutic T-cell applications is towards the QC, or manufacturing quality control, which allows one to optimize the production of the T-cells. Now, the second application is to monitor the activity of the therapeutic T-cells once they are introduced into an individual, asking questions like how long do the therapeutic T-cells persist? Are they expanding? These questions can provide insight into the efficacy of a product. So this slide highlights ways in which one can use TCR sequencing during the QC process. One can, at the very early steps when one is extracting T-cells from an individual for in vitro culture, one can assess the repertoire to understand whether it is diverse, whether there are dominating clones. During the in vitro expansion, one can monitor the expansion by assessing the T-cell repertoire features, again asking whether or not certain clones are dominating the culture. After a viral transduction step or other event that alters the genome of the T cells, it's important to assess whether or not such procedures may have given rise to T cells that are damaged and proliferating uncontrollably. So T cell receptor sequencing is great for that. And then finally, as I mentioned in the previous slide, when those T cells are introduced into an individual, the T cell receptor sequence can be used to track those T cells and ask whether or not they're expanding or whether they are no longer found in the individual. So just to get back to one important measurement of the repertoire, there's something called evenness. And repertoire evenness refers to the extent to which clones are, T cell clones are found in equal proportion. High evenness means that the clones found in a sample are all of similar frequencies, whereas low evenness indicates that some clones are preferentially expanded. And evenness is also known as normalized Shannon entropy. And for those who use clonality, it's 1 minus the clonality. So with respect to evenness, one great aspect of this assay is that one can quickly, within a 48-hour turnaround time, assess the repertoire. So start to sample collection to results can be obtained in under 48 hours. So in this experiment, we sought to track the evenness of T-cell therapeutic T-cells as part of a research study. These T-cells were derived from peripheral blood mononuclear cells. They were extracted and expanded in vitro using Dyna beads, anti-CD3, CD28 beads, and CTS optimizer. So these were expanded over a 10-day time course, and over the course of this in vitro expansion, we asked whether or not the evenness of clones changed. And in this type of experiment, one would hope to elicit a polyclonal T-cell expansion, where no one clone is dominating the culture. And indeed, for two donors, so two separate T-cell starting materials, we note a consistent increase in the evenness of the T-cells over time. So this highlights the fact that this approach with anti-CD3, CD28 bead stimulation and this optimizer media really promotes polyclonal or unbiased T-cell expansion. Now, in this last vignette, I want to mention one way in which this assay was used to provide insight into an adoptive T-cell transfer therapy. Now, the experiment I'm going to describe to you was presented by Dr. Noel Miranda, and in this study, an individual with cancer, there was a tumor extracted to create a tumor cell line, and T-cells extracted from this individual were co-cultured with the tumor. The goal there is to elicit or stimulate the proliferation of T-cells that are recognizing tumor antigen. After this in vitro stimulation of those T-cells, the resultant product is infused back into the individual with the goal of eliciting an anti-cancer response. So the individual in question had stage 4 melanoma and previously received adoptive T-cell transfer and lived for 11 years. But the cancer returned, and Dr. Miranda had to decide on one of two cell therapy products to give this individual. And at that time, they didn't use T-cell receptor sequencing to assess the quality of those cells. And so he really had to blindly choose which of two products to use. And you can ask yourself now, retrospectively, he went back and asked, what of those two products, what did the repertoires look like? And on the left, the two cell therapy products are indicated on the slide till 1207 and 1209. You'll note that there are quite striking differences in the level of T-cell expansion within these two products. The plots on this slide are spectra typing plots, which highlight clonal expansion, and they're taken directly from Ion Reporter. You'll note this 07 culture on the left contains two large circles indicating two large clones that are dominating that T-cell product, whereas the one on the right, many circles, a diverse product with many unique TCRs. Now, not knowing which the diversity of these samples, he chose the 07, and unfortunately, that individual had a negative outcome. And his argument, and which makes a lot of sense, is that perhaps the 1209 would have been a better choice because it's more diverse, and thus there are a greater number of TCRs that can recognize a greater number of tumor antigens. So this highlights the way in which this TCR sequencing approach can be used to improve or understand therapeutic T-cell applications. So with that, I want to finish and just summarize what I told you in this webinar. I talked to you about the Ion Gene Studio S5 system and how aspects of that sequencer, the Ion Torrent, really are well suited for immune repertoire sequencing applications, namely the low substitution error rate, long read capability, and fast turnaround time of this platform. I told you how long amplicon sequencing of beta chains from pretreatment peripheral blood allows for interrogation of polymorphism and also convergence to potentially predict response to immunotherapy and toxicity. And then I finally talked to you about ways in which that fast turnaround time and low input requirements allow the TCR beta LR assay to be used for therapeutic T-cell applications. So with that, I think we can turn to questions in the audience, and I really thank you for taking the time to listen to me.
Thank you, Dr. Looney, for that informative presentation. We will now start the live Q&A portion of the webinar. Again, if you have a question you would like to ask, please do so now. Just click on the Answer a Question box located on the far left of your screen. We'll try to answer as many questions as we have time for. Okay, let's get started. Dr. Looney, our first question is, how does the mutation rate of CA cells affect TCR convergence?
Could you of CA cells, could you help me understand better understand that term?
No, let's, Stephen, you've submitted this question. Would you mind submitting your question again with a little more elaboration on what you're asking for? We'll go ahead and skip this question, and hopefully Stephen will submit a more detailed question for you. Let's go to our next question. Okay, Dr. Looney, do you see convergent TCRB signatures in PBL, also in acute or chronic immune activation or disease?
Great question. This is something that we're actively exploring at this time. From the data we've collected, it does appear that one can detect some level of TCR convergence in peripheral blood of healthy individuals, but it's typically less than 1% of the total sequences. Now, in terms of chronic autoimmune disease, this is a great question, and one could hypothesize that convergence in this situation may be elevated. We don't know yet, but that's a great question and something that could be explored further.
Okay, we're going to go back to Stephen's question. How does the mutation rate of cancer cells affect TCR convergence?
Oh, great. This is, thanks, Stephen. That's a great question. And I think that, the broader question is, how does somatic mutation or the neoantigen load of the tumor correlate with convergence? We don't know yet. That's a great experiment. One could assess tumor mutation burden and ask whether or not it correlates with the extent of TCR convergence in peripheral blood. it's a really nice question, and it would be a great experiment.
Okay. Our next question. Dr. Looney, how can you accurately measure evenness or clinical expansion when starting from RNA? Every cell potentially has a different copy count.
Thanks. This is a great question, and this is something that I think is talked about in the field, the repertoire field, quite a bit. What are the advantages of RNA versus DNA? Now, DNA, it has to have one rearrangement copy per cell, whereas the RNA theoretically could have a different level of expression. Now, this question I would say is most pertinent to B cells, where the expression of the B cell receptor can vary tremendously depending on the B cell type. For example, memory B cell compared to plasmablast compared to plasma cell, the expression of the B cell receptor can vary considerably. And so that's something that one would need to take into account when looking at B cell receptors, given that a plasmablast or a plasma cell may have an outsized contribution to the RNA pool. Now, with T cells, there's really not, to my understanding, indication that different T cell subtypes have markedly different levels of expression of the T cell receptor. So that type of issue isn't as pressing for T cell studies. And the other thing I want to mention, which is a really nice aspect of the RNA approach, is that one can use quite a bit smaller amount of starting material in order to survey an equivalent number of T cell receptors that one would be able to capture through a gDNA-based approach. And that's because the RNA is already enriched for the target, the T cell receptor beta. So that's, I would just mention that's a really, really nice advantage of using RNA for this type of experiment.
Now, Dr. Looney, is this assay for liquid-based samples only, or can we use FFPE also?
Yeah, the LR assay is not it's not designed for FFPE, owing to the fact that the template molecules with the are often truncated, they're degraded owing to the FFPE preservation. So this is for the ideal sample types for this LR assay are peripheral blood leukocytes, whole blood, cultured T cells, and any fresh frozen biopsies. So really anything that is not FFPE preserved.
Now, Dr. Looney, this question next. Are you confident that TCRB signatures reflect the associated TCRA signatures that are not sequenced in the assay, given the preferential responsiveness of TCRB signals to exogenesis or endogenesis superantigen?
I want to mention, yeah, that's great. It's a great question. I think that one of the points that this question addresses is what does the alpha chain have to contribute to this? One question in the field is what proportion of TCRs are beta chain dominant, and that refers to beta T cell receptors where the antigen specificity of the receptor is wholly dictated by the beta chain, with the alpha chain having really more of a stabilization role. So in terms of the role of the alpha chain, that's not something we sequence in this assay. This is a beta chain sequencing, but one could make the argument that perhaps even a majority of TCRs are beta chain dominant, where any alpha chain paired with the beta chain would confer the same antigen specificity.
Okay, our next question. We have so many great questions coming in. Let's go with this one. Dr. Looney, a follow-up to the RNA versus DNA question, would you use RT-PCR convergence if you felt RNA was easier to use for T and B cell studies?
Would I use RT-PCR convergence? I think I'm still a little bit unsure of the meaning of that term. I mentioned that RNA in general is, I would say, it's really a preferential source of input material, owing to the fact that one can use a small amount of the material to survey a great number of T cells. So the other thing is that it's easy to obtain RNA usually from most samples, and the other benefit to using the RNA is that one will only sequence productive rearrangements. So that in some cases, T cells can have a productively rearranged TCR beta and also an unproductive. And with RNA, one will exclusively sequence the productive rearrangements. So I don't know if I have addressed that question, but if the asker would like to follow up with a little bit more detail of the RT-PCR, I'd be happy to answer.
Excellent. Now we have time for a few more questions. We'll try to get a couple more in. Let's go with, here we go. Dr. Looney, what is the read length of this assay?
Yeah, the amplicon length is 330 base pairs, and that covers the CDR1, 2, and 3. And this is obtained using framework 1 and constant gene targeting primers. Now the assay, those amplicons are sequenced with the S5-530 chip, and that chip has an up to 600 base pair sequencing length. So it's ample ability to sequence through this entire 330 on average base pair amplicon.
Okay, and Dr. Looney, we have time for one more question. Is this assay only compatible with the Ion 530 chip?
No, that's a good question, because it's essential for the long read lengths, but the 520 and the 510 are also certainly can be used. And the difference between those chips is just the output. So for the experiments I showed you, we used the 530. That allows one for typical use cases to have 8 samples multiplexed per chip at a level of 1.5 million raw reads. But certainly you could use those other chips.
Thank you, Dr. Looney, for your time today. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Dr. Looney, would you like to leave us with any final comments?
Yeah, I would. I'll just mention that today I talked to you about the LR assay, but in the upcoming webinar, we will focus on applications of our SR short read assay, CDR3 only, which allows for interrogation of FFPE treated material. It's going to be really nice. We'll have some exciting data to show you, and I would love to have you there for the next installment.
Thank you, Dr. Looney. Now, before we go, I'd like to thank our audience for joining us today and for your interesting questions. Now, any questions we did not have time for or those submitted during the on-demand period will be addressed by Dr. Looney via the contact information you provided at the time of your registration. At the end of this webinar, you will be redirected to the registration page for our third webinar in our 3-part immuno-oncology series titled Advancing Precision Immunotherapy Through Next Generation Sequencing of T-Cell Receptors, presented again by Dr. Timothy Looney. This live webinar is on September 12th at 8 a.m. Pacific Time. This webcast can be viewed on demand through February of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. We hope to see you on September 12th. Until next time, goodbye.